Let's have a look on the extracellular matrix components. First, we'll deal with the fibers. The most common fibers of the extracellular matrix are made of collagen. There's a wide range of collagen types, more than 25 types. However, we deal mostly with the type 1 collagen in this first look. And the collagen is produced mainly by fibroblasts, might be produced by smooth muscle cells as well. However, the point is then that the mother cells are producing precursors of collagen. So if this is a mother fibroblast, with the rough endoplasmic reticulum and the Golgi complex, even before the exocytosis, there are important processes such as hydroxylation of some coded amino acids such as proline, which is hydroxylated to hydroxyproline, or lysine, being hydroxylated to hydroxylysine. And these hydroxylated amino acids contribute to the stability of collagen fibers because they form many H bonds, stabilizing the integrity and the inner structure of the collagen fibers. It's interesting that the enzyme systems needed for regeneration of enzymes responsible for that, such as lysyl hydroxylase and prolyl hydroxylase, are depending on the vitamin C, without which we wouldn't have stable collagen fibers. So there's exocytosis of precursors, and let's consider one of these precursors, which is called procollagen. Procollagen. Let's mention the general structure of the collagen, where every third molecule is glycine. So this is general formula of collagen. It's glycine, then you have proline or hydroxyproline, and then you have another amino acid. And this pattern repeats again and again. The presence of the smallest amino acid, glycine, is responsible for a very close interaction between the three chains of procollagen. So it's a triple helix of three chains. And the chains might come pretty much close together, being stabilized by the H bonds due to the small glycine molecules. So this is a triple helix structure of the procollagen. On the end, they are the terminal peptides, which are removed, and in the next step you got just the triple helix without these end parts. So one protein chain, another one, and the third one. So this form is called tropocollagen. Tropocollagen. We have three protein chains stabilized by the H bonds. Each of these tropocollagen molecules then contributes to formation of structures of higher order. There is, if on a small scale, this would be a tropocollagen. These tropocollagen units periodically overlap with a constant period and while the length of each of the tropocollagen is approximately 300 nanometers, the overlap between the neighboring molecules is like 67 nanometers. So there is a periodic overlap of the tropocollagen molecules. Thus, the tropocollagen molecules form structures of higher order called collagen microfibrils, which have a cross-striation optical phenomenon. This results from the periodic overlap. So in electron microscope, you can see this cross-striation. And these structures of higher order that are like 20 to 100 nanometers thick are called collagen microfibrils. So each collagen microfibrils consists of a number of tropocollagen molecules periodically overlapping. However, these collagen microfibrils are organized again into structures of higher order. So if this would be individual microfibrils, they are organized into collagen fibers. So this is a collagen fiber. Which could have a diameter of approximately 1 to 2 micrometers. And the group of collagen fibers contributes to formation of larger structures called bundles of collagen fibers. Bundle of collagen fibers with a diameter approximately between 5 to 10 micrometers, and that is something that you can already pretty well see in the routine optical microscope. So these are the microfibrils at the beginning, okay? Another type of fibers you can find frequently in connective tissues are the reticular fibers. Reticular fibers or reticular microfibrils, because they are technically made of type 3 collagen that does not have this tendency to group into large and thick fibers and bundles. Therefore, the reticular microfibrils, which we have already illustrated in the context of reticular cells, are pretty thin, so we would not be able to see these in our routine sections. However, we can use their ability to reduce the atomic silver in a staining solution to black silver atom, so you reduce the cations, the silver cations, to black silver, which is the principle of classical photography, and that's why we call these fibers argyrophilic, and they appear as black fibers. stained black. Another type of fibers are the elastic fibers. Elastic fibers are compound systems made of a core of an amorphous protein called elastin. That's it. And this elastin core is surrounded with many microfibrils made of another protein called fibrillin. So we have the amorphous elastin core surrounded by microfibrils made of a protein called fibrillin. Elastin fibers contain domain made preferentially of hydrophobic amino acids, and therefore the elastin chains have a natural tendency to be organized in this more compact form. The elastin molecules are coupled or are connected by covalent bridges, which are the disulfidic bonds. So these are single elastic molecules with their hydrophobic domains with a natural tendency to minimize their contact with the surrounding hydrophilic tissue fluid, and we have these stabilizing bonds among the molecules, and that's how the elastin looks in the relaxed condition. However, once it's stretched by an external force, it's forced to straighten the chains a bit. But once it's given an opportunity to relax spontaneously, it returns the mechanical energy back and comes into that relaxed condition. So that's the principle. That's the molecular principle of elasticity of elastic fibers. Next to fibers, we should mention also the ground substance of connective tissues, which is made of glycosaminoglycans, which are basically made of repeating polysaccharide chains with various often modified saccharide molecules. However, they are repeating polysaccharide chains, etc. The point here is that due to the presence of hydrophilic groups substituting on these chains, they act as a polyanions in the tissues, being responsible for the hydrophilic properties of tissues. So they bind amounts of water. So they are extremely hydrophilic. They're responsible for binding the water in our tissues. Another group of molecules are the proteoglycans. And the structural principle is different. There is a protein core in the center with numbers of glycosaminoglycan chains on it. So it looks like some form of brush, right? So you have this protein core, but the majority of the whole molecular or supramolecular complex is made of sulfated glycosaminoglycans, approximately 90% or more, which is different principle than in glycoproteins. The glycoproteins are made of a protein, which is like a backbone of the whole molecule. It's often a globular protein. The surface of which is modified with oligosaccharides that are often branched. These glycoproteins very often have several adhesive domains, therefore binding the extracellular matrix to the cells, thus contributing to the cohesion and adhesion properties. So these are branched saccharide chains. So this is the basic overview. More details can be found in lectures and the textbooks, and good luck with that.